Everything you need to know about aspirin. Hello, my name's Rushil, and I'm here to give you a presentation on aspirin. First off, we'll start with background. This section will focus on the history and Canadian contributions of aspirin. What is aspirin? Aspirin is a non-steroidal anti-inflammatory drug, also known as acetylsalicylic acid. Aspirin was the first to be discovered of this class of drug. People have used aspirin as a common drug for relieving minor aches, pains, and fevers. It is also used as an anti-inflammatory or a blood thinner. There are different forms of aspirin that can be bought over the counter or prescribed by a doctor. Canadian involvement. First Nations people have long recognized the substances that make up aspirin and other painkillers effective. It is also present in several willow trees species native to Canada. Canadian scientists. In the early 1970s, when medical professionals had little understanding of the treatments and preventions of strokes, neurologist Henry Barnett oversaw groundbreaking research projects that upended conventional wisdom. Acetylsalicylic acid, ACA, which people have been using for 70 years to treat fevers, was shown to have the unforeseen benefit of preventing strokes in his first study. The now famous Canadian aspirin trial, the discovery provided physicians with a potent new therapy that is still often used today. A history of aspirin. Although aspirin's origins may be traced back to 1897, its intriguing history actually goes back more than 3,500 years, when Sumerians and Egyptians first employed willow bark as an antipyretic and painkiller, followed by famous doctors from ancient Greece and Rome. According to a Bayer timeline, chemist Felix Hoffmann utilized acetylsalicylic acid to treat his father's rheumatic in the late 1890s, which led to the development of aspirin we know today. Beginning in 1899, Bayer gave this ingredient-containing powder to doctors to deliver to patients. Economic impact. Aspirin is currently produced in excess of 40,000 tons annually throughout the world, and in the United States alone, more than 50 million individuals take 10 to 20 billion aspirin tablets to prevent cardiovascular diseases. Advancements. Aspirin started to alter patient and physician experiences and expectations, as well as the very structure of modern medicine, by offering a quick and affordable way to relieve pain. Prior to the middle of the 19th century, Western doctors viewed pain as a crucial diagnostic tool that aspirin helped to mask. Now physicians would have to focus on other symptoms. The second unit we will cover is biochemistry. This section will focus on the molecular structure and reactions of aspirin. The molecular structure. Aspirin has a molecular formula of C9H8O4. Functional groups. There's a carboxylic acid and ester group present. You can also see a benzene ring. When creating aspirin, the raw materials include phenol, sodium hydroxide, carbon dioxide, acetate anhydride, and hydrogen. The first step is creating aspirin is synthesizing phenol with sodium hydroxide to produce sodium phentate and water. The second step is adding CO2 to sodium phenate to create sodium salicylate. Third step is creating salicylic acid by synthesizing sodium salate plus hydrogen. The fourth and final step in preparing aspirin is by chemical synthesis from salicylic acid through acetylation with acetic anhydride, which will give you aspirin and acetic acid as a byproduct. Acetylation is a reaction that introduces a functional group, acetox group, CH3CO. It happens when acetox group is introduced in an organic chemical compound, usually for the substitution of acetyl group for a hydrogen atom. Reactions. Aspirin chemical reactivity, diagram A. Aspirin can easily enter cells and react with many different cellular chemicals. Example, aspirin will react with water to form the hydrolysis byproduct salicylic acid. Another reaction that can happen with aspirin is when it reacts with nucleic metabolites, example glutathione, or proteins to produce acylate products. A nucleophile is a reactant which gives an electron pair to form a covalent bond. In biochemistry, a metabolite is an intermediate or end product of metabolism. Diagram B. Aspirin is also capable of binding to the enzyme active site and modifying nucleic functional groups. Example, cyclooxygenase, COX-1, aspirin acetate, CYR530, rendering the enzyme inactive. Diagram C. This is a schematic to better understand the sites of acetylation on aspirin. In this experiment, the active site of aspirin acetylates is nucleic amino acid. Then the pools of protein is incubated with probes to reveal catalytically inactive functional groups within the now dead enzyme. In chemistry, a catalytic substance or a substance with catalytic properties is a substance which increases the speed of a chemical reaction. What type of linkage is present in aspirin? Aspirin is a dipole molecule with dipole-dipole attraction bonds and an hydroxyl segment as part of a carboxylic acid group. It has a covalent bond, which means electrons are shared. What properties does aspirin have? Properties of aspirin include being hydrophilic, polar, analgesic, anti-inflammatory, antipyretic, antiplatelet, anti-cancer. Analgesic is a pain reliever or a painkiller. is any member of the group of drugs used to achieve relief from pain. Antipyretic is a substance that reduces fever. Antipyretic causes the hypothalamus to override a prostaglandin-induced increase in temperature. The body then works to lower the temperature, which results in a reduction in fever. Anti-inflammatory is the property of a substance or treatment that reduces inflammation or swelling. What properties does aspirin have? Aspirin, or acetylsalicylic acid, is a colorless to white crystalline solid. It smells similar to vinegar. The smell comes from the hydrolysis reaction yielding salicylic and acetic acid of acetylsalicylic acid. Additionally, it has a bitter taste and will also decompose at high temperatures, melting point being 135 degrees Celsius. Is aspirin actively or passively transported? When ingested orally, acetylsalicylic acid is rapidly absorbed in both the stomach and proximal small intestine. The non-ionized acetylsalicylic acid passes through the stomach lining by passive diffusion. Aspirin is a prodrug which is transformed into salicylate in the stomach, in the intestinal mucus, in the blood, and mainly in the liver. Passive diffusion is a process by which molecules diffuse from a region of higher concentration to a region of lower concentration. It is the most important mechanism of passage of drugs through membranes. Homeostasis. This section will focus on how aspirin interacts with the many systems involved in homeostasis. For example, the nervous system, endocrine system, and excretory system. How does aspirin interact with the nervous system? When your nerves transmit an electrical signal to your brain, you experience pain. When you suffer an injury, the damaged tissue releases prostaglandins, which are molecules that act as hormones and induce the tissue to swell. They also make the electrical signal from the nerve stronger, which makes the pain you experience worse. Aspirin belongs to the non-steroidal anti-inflammatory drug class. By inhibiting the action of specific enzymes that produce prostaglandins, aspirin and other non-steroidal anti-inflammatory drugs are frequently used to treat these nerve signals chemically. Not only does aspirin chemically block your body's pain signals, but it also lowers your chances of heart attack and severe types of stroke. By increasing blood flow to your heart and brain, aspirin lowers the risk by preventing platelets in your blood from clumping and clotting in your arteries. The only over-the-counter painkiller with these potential life-saving properties is aspirin. Less discomfort and swelling result from this due to its effect as an analgesic, pain reliever, an antipyretic, fever reducer, and an anti-inflammatory. Aspirin helps to minimize discomfort and swelling, fighting swelling and inflammation. How does aspirin interact with the endocrine system? Aspirin interferes its acetyl group to one of the amino acid residues in the active site of the enzyme cyclooxygenase, or COX for short. To be specific, it's acetylserine 529 in COX-1 and serine 516 in COX-2. These are homologous serine residues, meaning they are the same relative position in the active site and perform the same function. In the image, you can see the oxygen atom being attacked by the carbonyl on aspirin and is thereby acetylated, converting aspirin into salicylic acid in the process. So by altering the structure of the active site, aspirin renders the enzyme permanently ineffective. This means new enzymes must be made in the body before the effects of aspirin wear off. How does aspirin interact with the excretory system? Salicylates are primarily excreted from the body through the kidneys, through the glomerular filtration and tubular excretion process as free salicylic acid, salicylic acid, as well as phenolic and acylic glucuronides label. Shortly after administration, salicylate can be detected in the urine, but it takes around 48 hours for the entire dose to be entirely excreted. Salicylate levels in urine can range from 10% to 85%, and they are highly influenced by the pH of the urine. While alkaline urine tends to promote excretion, acid urine typically helps the renal tubules reabsorb salicylate. What biofeedback does aspirin cause, positive or negative? Aspirin causes a negative feedback response in the body as it irreversibly acetylates and inactivates COX-1 and COX-2 enzymes. As the liver and brain are actively trying to get rid of aspirin, it brings the pain and inflammatory responses back to a normal range. Does it affect urinalysis? When aspirin is taken in high dosage, it tends to increase uric acid excretion. When aspirin is taken in lower dosage, it may cause uric acid retention. Disorders that happen with taking aspirin may be Reye's syndrome. Despite its many uses, certain patients can display a hypersensitivity to aspirin. 10 to 25% of people with asthma have their symptoms worsened by use of aspirin. Additionally, aspirin has been associated with Reye's syndrome, which is characterized by swelling of the brain and liver. It is most common in young children, and that is why aspirin is not given to children or young adults. One more thing to note before starting the next section is because of how aspirin irreversibly stops the COX enzyme, it makes it very effective at low dosage to prevent blood clotting. Section 4, Genetics. This section will focus on how people have built up genetic tolerance to aspirin. Genetic resistance. Despite the clinical advantages of aspirin, there is a significant inter-individual variation in how people respond to the antiplatelet medication. Aspirin resistance, AR, is regularly seen in action, but we still don't fully understand how it works, although the cause of AR are likely to have multiple genetic factors that seem to prevail. Numerous single nucleotide polymorphism in genes associated to cyclooxygenase thromboxism and platelet receptors have been found as being capable of negatively influencing aspirin action. According to a number of genetic association studies, including genome-wide and Canadian gene studies. Because they may be crucial in establishing the AR phenotype, it is essential to know the clinical implications of AR-related SNPs as possible predictive and prognosis biomarkers. Section 5, Metabolic Processes, is our last section. This section will focus on how aspirin affects different metabolic processes, such as glycolysis cycle, electron transport chain, and pyruvate cycle. Starting off with glycolysis. Aspirin treatment of human platelets reduce the concentration of adenosine triphosphate and hinder glycolysis. Adenosine diphosphate was released along with an increase in glycolysis when collagen was exposed to platelets. While aspirin pre-incubates of platelets for 5 minutes entirely prevented the release of adenosine diphosphate in response to collagen exposure, it somewhat suppressed the rise in glycolysis following collagen exposure. Release of adenosine diphosphate and the transport of glucose through the platelet membrane are thought to be inhibited by aspirin when it interacts with human platelets. Next, pyruvate. Aspirin-treated mouse embryonic fibroblasts displayed a general decrease in oxygen consumption in response to the carbohydrate substrates pyruvate and succinates, which is consistent with increased mitochondrial fragmentation. The mitochondrial network is disrupted by aspirin, and respiration on pyruvate and succinate is decreased. Electron transport chain. It is unclear how salicylates affect the metabolism. In 2 separate cell lines, aspirin boosted long-chain fatty acid oxidation in the mitochondria, but decreased peroxisomal fatty acid oxidation. Aspirin boosted the acetylation of mitochondrial proteins and was found to be a stronger in vitro acetylating agent than acetyl coenzyme. All of these findings suggest that aspirin likely has its effects without directly affecting the fatty acid oxidation enzyme in the mitochondrial matrix. Instead, it likely affects how long-chain fatty acids are transported into the mitochondria. The increase in mitochondrial fatty acid oxidation may be a response to the changed mitochondrial shape and impaired electron transport chain activity, both of which were noticed after cells were exposed to aspirin for 24 hours. These findings shed light on the pathophysiology of Reye syndrome, which is known to be brought on by fatty acid oxidation abnormalities in patients using aspirin. With that, I would like to end my presentation on aspirin. Thank you for listening.